Good afternoon and welcome everyone to the 2024 PI Conference and our 3:30 session, Gene Editing for XLA. My name is Emma Mertens, Program Manager of Community Health at the Immune Deficiency Foundation, and I will be your moderator for this session. This session is 45 minutes in length, and if time permits, Dr. Cohn will be happy to address a handful of attendee questions after his talk. Please note that the information presented during this session is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with questions concerning your medical condition. Here at IDF, we offer a wide array of educational presentations, including those developed by healthcare and lifestyle management professionals who have been invited to serve as speakers at this conference. The views and opinions expressed by guest speakers do not necessarily reflect the views and opinions of IDF. But without further ado, I am so proud to introduce our presenter, Dr. Donald Cohn. Dr. Cohn is a distinguished professor in the departments of microbiology, immunology, and molecular genetics, pediatric hematology and oncology, and molecular and medical pharmacology at the University of California, Los Angeles. He is the recipient of IDF's 2023 Michael Blaze Research Grant Award and is here today to share on his work focusing on gene editing for XLA. For full presenter bios, please visit your conference app, and welcome Dr. Cohn.
Well, good afternoon. It's getting to be kind of a long day. I gave a talk about 6 hours ago, but I'm still fresh. So I'm going to talk about gene therapy for XLA. I'll give a little background on gene therapy, although I gave a talk this morning also that covered it more. And I have to, my disclaimer is I'm a bone marrow transplant physician, so I don't actually take care of patients with XLA because they generally don't get transplants. So if you have questions about IgG replacement, I'm not the person. But you're welcome to ask any questions you have. So this is our UCLA beautiful campus. So this is the original report on agammaglobulinemia. So Colonel Ogden Bruton, who was an Army pediatrician, described a patient who had a complete absence of gamma globulin in the human serum. And this patient had several years of many infections. And in fact, he gave the patient gamma globulin to control the infections. This was from 1952. It's a picture of the colonel. And this is the electronic medical record of the day. So this is his data on the patient. So going down, so it's 1946 to 52. Going down the left, each one of those dots is the time the patient had sepsis, bacteria in their blood. The next column is which types of pneumococcus they were able to identify in some of those infections. Then a list of other infections. So they had rubeola or German measles, varicella or chickenpox. They had epidemic parotitis, which I think means mumps. And they actually had that 3 times. And once you have mumps once, you should make enough antibodies not to be able to get that again. And other infections. Then on the right are the treatments. So they did the standard thing in those days, TNA tonsillectomies and adenoidectomy. I had that around that age, a few years after that. Then they gave them a vaccine. They tried sulfa. They x-rayed their nasopharynx. They tried other immunizations. And then the last thing he did was give him gamma globulin. And after that time point, there's no more infections for the year of follow-up. And the diagnostic was, again, sort of, nowadays we do a whole exome sequencing. So this is serum protein electrophoresis. You just take some of the liquid of the blood and run it in an electrical current. And you see across the top is from the healthy donor. You see the big peak is albumin, which is the major protein in the blood. Then there's all those little peaks that are labeled alpha, beta, gamma, delta. The middle tracing is the patient. And you see there's no gamma tracing. So we recognize this patient with multiple infections didn't have gamma globulin in their blood. And the bottom one is after he started giving the patient gamma globulin. You can see there's a little rise above the background. And in fact, probably the amount he was giving him was a few percent of what a patient would get nowadays with modern gamma globulin. So the gamma globulin preparations that we give now, we're able to give so much more than we could when you're giving either a little bit. He was actually giving it sub-Q, but for many years it was IM gamma globulin. You're getting very little. but it was enough to change his course as you saw that he stopped having infections. So XLA, it was learned, the first patient was male, but after multiple patients were observed, it was clear that it was affecting males. So it was the first clinical characterization of a primary immune deficiency, and the estimated incidence is about 1 in 200,000, meaning about 1 out of 100,000 boys. And it's caused by mutations in a gene that, when it was identified, was named by the discoverers after Colonel Bruton, so it's called Bruton's tyrosine kinase. Kinases are enzymes that usually are involved in cell signaling and cell growth signaling. And it turns out, as I'll show you, this one's essential for B cells. And so when we make from stem cells, we make mature B cells on the far right. They grow through various stages and they express different proteins. And sort of in the middle of this process, the BTK gene turns on and it gives the cells a signal to keep growing. And if you are missing BTK because you inherited a defective copy of it and you have XLA, you only make very early B cells and they stop at that point. So you don't make mature B cells, so you can't make antibodies, hence the absence of gamma globulin or agammaglobulinemia. And so it's an inborn error of immunity with absent mature B cells and immunoglobulins. Patients don't make antibodies and therefore are treated with immunoglobulin supplementation. And this treatment, as many of you know, is expensive and lifelong. And even despite effective immunoglobulin replacement, patients still can have ongoing infections. The immunoglobulin replacement is IgG. It doesn't replace, for example, IgA, which we make sort of in our nasopharynx and our mucosa, and so they can still have infections, and rarely severe viral infections. So we know that, in fact, if you look at different patients with XLA, they have different mutations in the BTK genes. There's multiple different ways to make the gene not work. And if we want to replace it, if we want to give the patients a normal copy of it, we need to make sure that it's expressed exactly like the normal BTK gene is, that it's off till that certain point, it comes on, and then it's well controlled throughout B cell development. So you can't really just put it in sort of in a stuck-on position like we can with other genes. So one of the enzymes, one of the diseases I work with is ADA-SCID, and there the gene ADA is for an enzyme that every cell in the body makes. If you have a little, it's enough. It's hard to have too much. So you can just put it in with a virus and express it, and it's okay. But BTK really needs precise regulation to be effective and safe. And so I showed this slide earlier. This is just sort of a cartoon of gene therapy. And so if you have a gene or you have editing reagents, you can either give them into the body in vivo, either in a viral vector like AAV is a virus that's used often, or editing reagents, and you can, for example, inject that into the back of the eye, put it into muscle, into nerve cells, into liver cells, and they can make the protein. But for the immune deficiencies, we need to get it into the hematopoietic stem cell. Oops, let me back that up. So we need to, on the left side, the hematopoietic stem cell, which is the cell that makes all the blood cells. And then just a couple of other, I have different versions of this slide, so I forgot what was there. So a new gene transfer technique that's just sort of emerging are lipid nanoparticles. So the same material that was used to deliver the COVID vaccine is now being looked at as a way of delivering genes right in the body. It's not there yet, but it's coming along. And then one of the points to make is the gene therapy we're talking about is only to affect the cells in the body, but not the cells that get passed on to the next generation. So we're not going to get the gene into the sperm-forming or egg cells, so it only treats that one patient, but they still are carriers and still could pass it along, because we don't think we're wise enough yet, at least we're wise enough to know this, to start affecting the human gene pool. Because if we put a gene into someone's egg, for example, that could get passed on for generations forever. And so when we do it out of the body, as we're doing with stem cells, there's no chance of getting it into those other cells. So we're targeting stem cells. And so the idea is that in our bone marrow, we have these cells, as shown on the left, the hematopoietic stem cell, or hematopoietic is blood forming, that throughout our life self-renew. So they divide, they make more stem cells. So we have this permanent pool of stem cells for our whole life. But then some subset of them go on and move to the right and differentiate, specialize, and become either the blood cells shown at the top or the ones on the bottom. And on the bottom, you can see the bottom one is the B cells. And so all these different blood cells that we have in our blood on your far right all come from the stem cells. And so if we want to make a permanent change in the patient's blood-forming cells, we have to either transplant stem cells or fix the stem cells. So we have two main ways to do that. And one is to add a normal copy of the gene. So we can, for example, take a BTK gene that's been cloned, put it into a virus, and use that to add a copy to the stem cell, and then it would be in all the cells that come from there. Or now a newer set of technologies called gene editing, using something called CRISPR that I think I will explain for a bit, that will actually change the gene right in the stem cell. But we need to do it in the stem cell to have it to be permanent. And then this is just a marker. There's a protein called CD34 that's on about 1% of the cells in bone marrow. So a lot of times when we're processing cells from a patient, we'll collect their bone marrow and then isolate those that just have CD34 on the surface, so we just have less cells to work with, and we can sort of get rid of the other ones that are mature and aren't really what we're trying to target. And so this sort of cartoons the process of gene therapy. We start with a patient, and then we collect their stem cells either by taking it out of their bone marrow directly or chasing the cells into the bloodstream and drawing blood and collecting enough in a process called leukapheresis so that we can get a lot of the patient's blood-forming stem cells in the laboratory. And then we can, from the top, have a virus that we've cloned our gene, in this case BTK, into the virus and use that to infect the gene into the chromosome of the stem cell. So the virus that we use, lentivirus, is actually HIV. Lenti is its family of viruses. And HIV normally infects a cell and puts its DNA into the DNA of the stem cell. So our virus, our vectors, all the virus genes are gone. So it can't cause an immune deficiency. And we've been using these now in patients for 20 years and it's never caused a problem. But they're very efficient at getting into the cells and putting the DNA into the chromosome so that every time that cell divides, then it will pass it along. The newer approaches are shown at the bottom, which is gene editing, where we can use an enzyme called CRISPR to actually make changes at one specific site in the DNA to do what we want, and that's what we'll talk about mostly today. Then once that's done, then the patient will typically get some chemotherapy to wipe out the rest of the bone marrow in their body. So when we give back the cells that we've treated, they grow back the blood system. So over the course of about a month, the patient will regrow their blood system, but now all the cells will have the gene that we put into the stem cell. So if it was BTK, now the B cells have the information they need to make mature B cells and antibodies. So the first approach, using a viral vector, has been explored. Not a lot, but so the normal BTK gene was cloned. So they took blood cells from a person and actually isolated a normal copy of the gene in 1993. And about a decade later, David Rawlings at Seattle Children's reported putting that gene into a retrovirus and using it to put it into cells from XLA patients and correcting the defect. The field in general moved from mouse retro to HIV-based lentis because they're actually safer. So David's lab, again, had multiple papers with a lentivirus carrying a normal BTK gene and showing in either in the mouse model or in patient cells that adding the gene could correct the defect so the cells could start making B cells. Could start making B cells. And then also Frank Stahl in the Netherlands, and actually misspelled his name, it's AAL, also had one paper with a lentiviral vector. So there have been multiple publications demonstrating that BTK gene transfer into stem cells with retro or lentiviral vectors restored B cell development and antibody production in both mouse models and in XLA patient cells in culture. However, I talked earlier about the importance of getting it regulated precisely, and it's not clear how precisely regulated it is. So David's group used a promoter that would have the gene only be on in B cells, but kind of all the time in B cells at a fixed level. I don't think any of the papers that were done reported seeing complications, but if you want to treat a patient with a therapy like this, the FDA will require a lot of testing to make sure that the virus doesn't cause problems. And so I'm not sure where that all stands if they're still going forward with it. But I think a lot of the work in the field is looking at this CRISPR approach to try and insert a gene in the right place. And so this is a map of the BTK gene. So a lot of our genes are built as shown here. The blue boxes are the part that codes for parts of the protein. Those are the exons. And the little dashed lines in between are the introns, intervening sequences. So our genes actually are not all right next to each other. The pieces are broken up. And if any of you have heard about whole exome sequencing, that would be just sequencing the blue parts but not the gray parts in between. Turns out that most of our genome is like the gray parts in between, and so it's easy to sequence the 2% that's exomes. So the BTK gene is broken up into 19 coding regions, and it turns out when different patients with BTK have had studies done to see what's their mutation in BTK, we found mutations all across the gene. So the gene is very sensitive. A change in almost anywhere in the gene will make it not work. And so it's going to be difficult to use like a technique to fix each individual mutation, although that may come in the future. And so the approach that we're looking at and other labs are looking at is to integrate a whole copy of the BTK gene. So that's just all the blue parts strung together early in the gene, so it should be effective no matter for any mutations downstream. So if we could take all those blue parts, string them together, and stick them between the bottom between 1 and 2, it'll express that protein. It doesn't matter what mutations the patients have downstream. And I'll show you another map of that because it's a little complicated. So gene editing is the approach that's being looked at. And the approach is based on this molecule called CRISPR. So now about 11 years ago, Jennifer Doudna and Emmanuel Charpentier discovered that bacteria have this system where they can actually defend themselves against an incoming virus. And so they have this protein Cas9, sort of the green outline thing. And if you have the yellow sequence, which is an RNA, you can design it so it matches a specific part of the DNA, and it will bind there, bring in that green protein, which cuts the DNA, the two little scissors. So you can actually target this to, out of the whole 3 billion bases, go to one place in the whole genome and make a cut in the DNA. And so what does that get you? So this is another version of that. So at the top is the patient's gene. M is a mutation. So this would be a mutation, for example, in the BTK gene. Well, there's a series of different enzymes that have been developed, with CRISPR being the newest and the winner and champion, that can bind to a single site in the DNA and make a double-stranded break. So cut the DNA there. So if you make that break and you don't add anything else going to your lower left, the cell will very quickly get those ends of DNA back together. When DNA breaks, it's like call the paramedics 911. And so there's a pathway to get the DNA back together, but it often takes out or adds a few letters at that joint. So that will disrupt the gene. So that's not what we want to do here. But that is useful if you want to knock out a gene. And so people have done that to like T cells to knock out the co-receptor for HIV. Or there's in fact now an approved gene therapy for sickle cell where they use this approach to knock out the repressor of fetal globin, so it turns on fetal globin, which can help sickle cell disease. The approach in the middle, if you add a piece of DNA that matches that cut site, but it's got the correct base, the normal sequence, the cell can use that to fix the break and knock in the normal sequence. So we're looking at that for sickle cell disease, where every patient has the exact same single base that's wrong to try and fix it. What we're going to talk about for BTK is the one on the right with the blue. So you can actually take a whole copy of a gene, like the blue would be a BTK gene, put next to it or flank it with the sequences of the cut site, and that will go into the cut site and introduce the gene at a specific place. And I'll show you another version of that here. So if this is the BTK gene, let's say this patient has a mutation in exon 2. We can make a synthetic version of all the coding information, all the exons for BTK, string them together, and flank them with sequences that match where we're going to cut. So then we made a CRISPR that cuts right there, makes a double-stranded break, and then through this process of homologous recombination, that piece of DNA will insert at the break. So now you have a normal copy of the BTK gene right next to its normal promoter that controls its expression. So you've put in a normal copy in the right place, so it will be expressed precisely like the normal gene would. And so that's the method that we hope will be effective to giving expression of the normal gene at the right time and place as B cells develop, because they're being controlled by the BTK control elements. And so here's just another version of it. So we have CRISPRs that cut around exon 2, for example, We have a whole version of everything past that will get inserted and get expressed from there, and you'll make BTK protein at the right time and place. And so then this is sort of just another important fact about XLA. So it turns out there's a strong selective advantage for B cell production from BTK replete, or stem cells that have BTK. And this was shown beautifully by studies done by Mary Ellen Connolly at St. Jude 25 years ago, I guess it is. So she has a mouse model that has XLA, basically. And if she could transplant into it as little as 0.5 to 5% of normal bone marrow stem cells, B cell function would be restored. So 0.5 to 5% bone marrow stem cells from wild-type donors, normal mice, restored antibody production, indicating that engrafting 5 to 10% of BTK gene-corrected stem cells may be therapeutic for XLA patients. So that means we don't have to fix all their stem cells. We can just give them about 5 to 10% of their stem cells that are fixed. That could be enough to make antibodies. So the selective advantage for B lineage progenitors from a relatively low percentage of gene-corrected stem cells may also allow reduced intensity conditioning, so much lower amounts of chemotherapy we might need to give for a regular transplant, as we use for SCID. We use about 1/4 of one chemotherapy drug. It's enough for SCID, and still achieve sufficient engraftment of gene-corrected stem cells to support production of B cells and protective antibodies. And so autologous transplant, patients' own stem cells for gene therapy may be important, potentially curative treatment with benefits exceeding those of lifelong immunoglobulin replacement. That's a hypothesis that will be tested when there are clinical trials. And so I want to show you some work that a grad student in my lab, actually pop up this picture, Chris Luthers is a senior grad student who's been doing these studies over the last couple of years. There's a mouse model that has mutations in the BTK and another gene that has very much like human XLA, so it makes no antibodies. So he took bone marrow from one of those mice that in the laboratory the next day treated it with CRISPR and a normal BTK gene, so he corrected them, and then he transplanted those into BTK knockout mice, and he put males into females so he could measure how much of the donor engrafted. So that's sort of the gene therapy into the mice part, so it's just like how we would do it for humans. And then we followed the mice over 20 weeks. So he drew blood samples in 8 and 12 weeks. Then at 15 weeks, he vaccinated them with an antigen, something they hadn't seen before, something called NPCGG. It's just a foreign substance that they can make antibodies to. And then he got some blood a week later. Then he reboosted them with the NPCGG at 19 weeks. Then at 20 weeks, euthanized the mice and then looked at their organs to see, what was the effect of the gene therapy. And so the first thing he measured was how many B cells they have. So he took blood samples from them, and by flow cytometry, he can measure how many B cells they have. And so the black bar is from a normal mouse, the number of B cells. They have 45% of their lymphocytes are B cells. The bar let's start from the left. So the low gray bar are these BTK knockout mice untreated. So they only have about 10% B cells, and if you look, they're actually immature B cells. The orangey bar, the dark orange bar, are BTK mice that got transplanted with normal bone marrow. So they don't quite reach the level of B cells as the normal mouse, but they do pretty well. And then the lighter orange bar are mice that got transplanted with uncorrected cells. So you can see they're like the mice that never got transplanted. Then the next two bars, the purple and the green, are mice that got the gene-edited marrow. And they are now making as many B cells as the mice that got normal bone marrow. So it says that by correcting them, they function normally. And when we measured, I don't have the data here, but about 15% of the stem cells in those mice have the corrective gene we put in. And that's enough for them to make B cells as well as if they got marrow from a normal mouse. Oops. And then he also measured IgG antibody production, and you see the same picture that on the left, the untransplanted mouse, or the light orange, the one that got uncorrected marrow, have very low levels of IgG, and those that got the edited cells on the two bars on the right were as good as the one that got wild-type marrow. So they're making antibodies after the gene therapy. And then this is from the vaccination. So this is measuring the levels of antibody to that foreign substance he gave. And so the three bars at the bottom are the mice that got uncorrected cells. The other ones are the ones that got either the normal cells or corrected cells. You can see initial response with the first vaccination and then a boost with the second vaccination. So they're having normal responses to vaccinations, which we often use clinically as a diagnostic test to say that patients' B cells are working because they can respond to vaccinations. And so as little as 15% correction made the B cells, the immunoglobulin, and the antibody response to the vaccination normal. So we think this is a very hopeful result to show that this approach can be beneficial. And so we're just in the end of wrapping that up. And now a postdoc in my lab, Aurelian Colamartino, is doing studies in cells from patients. And so in many ways, I'm fortunate to live in California, one of which is we have the California Institute for Regenerative Medicine. That's our stem cell agency that funds research. And they give out their grants sort of to do the different steps you need to do to get to a clinical trial. So we have a new grant, a Translational 1 grant, to get one step before the trial. And their grants are based on milestones, and you don't get the next amount of money till you reach the milestone, which is very smart from them. And so this is what we're planning to do to get to talk to the FDA to start our trial. So we've defined what's the optimal method for editing human cells, what's the concentration of reagents that works well, what's the time and culture. And so we're just now making one large-scale lot of cells from a healthy donor to show we can get the gene into them at a high percentage. And then we freeze the cells when we're done treating them. And one of the things you have to show the FDA is how long they're stable for. So you know how your Tylenol bottle says good until 2026. They've done stability testing and know that once they make it, it's good for 2 or 3 years. So we have to know that for our cells. So if we make cells for a patient and there's a delay in treating them, we know how long they're still good for. So that takes like a year to do. So we've kicked that off already. And then we're going to do studies in cells from XLA patients. And we have to test, is there any toxicity from doing this? So we'll treat human cells, put them into immune-deficient mice, let them grow for 4 months and make sure they don't grow any tumors or leukemias or any other abnormalities. We have to develop a clinical trial synopsis, which I'll show you on the next slide. And then we'll meet with FDA to get their sort of final instructions for what they want to see for our application. Once we've done that, we then apply for the next grant, and we have to do the next steps, which is do all the things the FDA told us at that meeting, including how we manufacture the product, the chemistry, manufacturing controls, whatever toxicology, or whatever else they want us to change in the clinical protocol. And once that's all done, we can then submit to the FDA an IND, which is an investigational new drug application, which is what you need from the FDA to give a patient an unapproved drug. And so these are the hoops we have to go to. And you know, it's a lot of work, but it's FDA's way of keeping us safe and making sure that when things go into clinical trial, there's a lot of information to say this can work and this should be safe. So we'd submit it, and then if the FDA accepts it, then the trial would open. And so 2 more CIRM grants to apply for along the way. The CLIN 1 that pays for those work and the CLIN 2 that would pay for a trial. And CIRM is very big on Gantt charts. I've gotten very good at making Gantt charts to lay out projects. They're very project-oriented, and so this is sort of the graphic version of what I talked about before, the various steps we have to do for pharmacology, toxicology, chemistry, manufacturing, controls, how we make the cells, clinical trial, and all the regulatory steps we have to go through to get from where we are now to a clinical trial. And so we hope that we can do it over 2 years optimistically. And so in thinking about what a clinical trial would look like, you know, there are trials, phase 1, 2, and 3. Phase 1 is usually the first time the drug has been given to people. But for gene therapy, we often combine 1 and 2. So it's the first time we're giving it to patients, so we're looking for safety issues. We also want to see some sign that it works, and that's the phase 2 part. So we can do that at the same time. We measure safety parameters, but also does it work? And so it would be autologous, the patient's own CD34 positive stem and progenitor cells after ex vivo targeted BTK gene insertions, the way I showed you that we would do that. And so from the FDA parlance, our test article, the drug that's being tested, are the autologous stem cells with a BTK gene insertion for the treatment of XLA. Trying to decide which patients would be eligible. They would have to have proven agammaglobulinemia, so blood tests showing before treatment they had very low levels of antibodies. We have to define that they have a mutation in the BTK gene that would inactivate it. They have to be well enough to have an autologous transplant, meaning heart, lungs, kidney, et cetera. And then typically the FDA wants us to start with adults if we can. So for SCID, there are not adults untreated with SCID, so the FDA allows us to start those trials in children. But if there is an adult population, because adults can give legal consent, typically you would start with adults, and then, for example, after 3 adult patients are treated and show safety and efficacy, you could then open the next phase of the trial and move, like treat adolescents. If that works well, you could move down. And so it would probably be a progressive decrease in the age of the study. And then this is just how we'd get our stem cells. We mobilize peripheral blood, selected for CD34 with the gene inserted, and presumably with reduced intensity conditioning, lower amounts of chemotherapy than you might give for a transplant from a donor. And so that's what we've been working on. That's already about 3 years of work, and we probably have another 2 years to get it into clinic. There are other people, I believe, pursuing the same approach, and may the best group win. May the patients win. I just want to mention that I think this is the last slide. There are newer methods coming along that are going to, the field is moving very rapidly. So something that's come along is base editing, where you don't cut the DNA, you can actually just change a single letter. So if the patient's mutation is a letter that it should be a G, but it's an A, there's a way to actually use base editing and change that A back to a G. And they turn out to be very efficient. I'll show you an example of it. Or there's another new method called prime editing, where you can actually write in a little sequence. So if a patient's missing a little bit of DNA, you can actually write it in. And so these are just moving into clinic now, but I think they're very precise, refined techniques, and I think they will become what we're using in a few years. So I showed you that map of different mutations in BTK. More than 600 different mutations have been described in BTK across different patients. And so these methods, base editing and prime editing, only work for a small region. So you'd have to make a different set of reagents, like that guide RNA, to bring it to different sites for each one. So it would be not straightforward, but I think there are high throughput methods where you could do that. And the FDA has recently proposed a platform approval approach, where you could get approved to use base editing for one mutation. And once you have that approval, it's much easier to add it to treat the next mutation, easier to do the next one. So you might be able to really quickly develop a whole sort of portfolio of editing techniques to treat all the different mutations in BTK. And so this is an example. This isn't for BTK. Grace was an undergraduate in my lab who wanted to try base editing. And we found that in Canada, there's a large group of Mennonites that have a mutation that causes SCID, in a gene called CD3 delta. And so they have the sequence on the left, it says TGA, that says stop. So that breaks the protein at the wrong place. And it should be what's shown on the right, CGA. And so you can make a base editor that uses kind of that same CRISPR machinery, but it brings an enzyme to that site, in this case, adenosine deaminase. It'll change that A ultimately to a G, and then you wind up with the correct base sequence that's shown on the right. And it turns out this is very, very efficient. We get like 80% correction when we treat patient cells with it. So we're trying to move this forward for CD3 delta SCID, but the same approach can be used where you just target that machinery to the mutation in BTK. So I'll stop there. This is my lab group at the end of our semester this year. These are the people in my lab that do all the clinical trial work. These are our many collaborators, and these are our funding agents. So I'll stop there. I hope that was understandable. I'm glad to answer questions. Thank you.
All right, thank you so much, Dr. Cohn. That was wonderful. Before we transition to a quick Q&A, I just want to share a couple of IDF resources, and then we will get into Q&A. So if you leave today with any lingering or unanswered questions, please visit our Ask IDF resource. A member of our team will reach out to you directly to provide support or connect you to an appropriate resource. We can also help connect you with other members of the PI community through our virtual Get Connected groups. IDF Get Connected groups offer individuals diagnosed with PI and their family members the opportunity to connect, share experiences, and receive support in their local communities. Finally, please remember to complete your session survey through the conference app and let us know if you enjoyed this session. All right, now we have some time available for a quick Q&A. If you have a question, please raise your hand, and Angie in the back there will bring the mic over to you. And we do ask that you ensure all questions are related to this session and topic. I'll let you come back up here.
Thank you so much for sharing. Congratulations on the progress. You mentioned May the Patient Win. I have two questions for you. One, to your knowledge, how many other teams are competing in the similar research?
You know, it's hard to know for sure until people either make presentations at meetings or publish. And I don't know that anyone else has published on BTK so far. I think David Rawlings' group has been working on this approach for a while in Seattle, and I'm not sure who else is.
And then if I may, at one point in your presentation, you mentioned something related in terms of addressing sickle cell, and I was just curious to what extent the method that you're researching, to what extent do you have a hypothesis that it may introduce a remedy for things in addition to XLA? Right.
So the sickle cell approach was basically just to cut and break a gene. It's very easy to break things, as we know. Our approach is we cut, and we want the whole piece to move into that site, and that's a rarer event. So if you cut, probably 80–90% of the genes will get broken. If we cut and we have our piece to go in, we get 15 to 30% taking it up, and so it's lower. But if that worked, and we could do that consistently and have those cells engraft in the patient and keep that gene working, then there's many of the immune deficiency diseases and many other metabolic diseases. So it could work for a whole number of genetic blood cell diseases if it works. The question is, you know, things always or often work in mice. They don't always work when you bring them to patients. And so that's why, you know, until you do a clinical trial, you don't really know. The only, you know, and mice generally tend to be easier to cure. Many people have cured cancer in mice many times, and when they go to human trials, you know, it's more difficult. So that's why, you know, we're not completely 100% confident this is going to work, and ultimately we need enough information to convince the FDA to let us open a clinical trial.
Hi, Dr. Ken.
Hi, Barbara.
So when you use CRISPR, what mechanism brings the piece of DNA up into in line?
Yes. So the way we generate, so the viruses that we use, they're self-mobilizing. That's why they're so good. You add the virus to the cell culture and it gets in and does its thing. To get all the editing reagents into cells, we typically use a method called electroporation. So we have the cells in suspension of a solution of the DNA, and we give it a strong electrical current that drives them into the cell. Okay. And so the CRISPR reagents will go in and do their thing. Delivering the big piece of DNA, like the normal BTK gene, it doesn't go in that way very efficiently. And so this is actually one of the still areas of research. So you can put it into an adeno-associated viral vector, and that can get into the cell pretty efficiently, but it's somewhat toxic to the stem cells. You can put it into a non-integrating lentiviral vector, that can take it in. So we're still finding the best way to deliver the normal gene part. That's the hardest part of it.
So you can't use a vector with CRISPR at the same time then?
Well, you can, but like a lentiviral vector, if you just used a regular, it's going to integrate on its own. It's going to kind of ignore the CRISPR mechanism. So we can make a lentivirus that won't integrate on its own, and that could provide the sequence to the CRISPR site. So, and that works fairly well. So we're sort of working between those two methods of getting a normal BTK gene. That winds up being a fairly big, relatively piece of DNA.
Yeah.
And so it needs to go in like typically in a virus, we think.
Okay.
And so that's an area of research, I think, across the field of how do we do that step. And I don't think anyone has the perfect answer.
Okay. Thanks. Thank you.
If I understood the timeline correctly, it sounds like the hope would be in about 24 months that you'd be able to potentially get FDA approval for clinical trials.
Right.
Is that correct?
That's
and
our
then, do you have an estimate of ones that went to clinical trials? Would that be 2 years, a decade, or is it really too hard to
You mean to get it FDA approved as a drug? Mm-hmm. So that's a difficult question. To get FDA approval of a drug, you need to demonstrate that you can make that drug under pharmaceutical conditions reproducibly. And so if a company is going to market Tylenol, they might make 10, 15 batches of Tylenol, one after another, and measure the properties of those and show all that information to FDA to demonstrate to them they can make the drug consistently reproducibly. To do that with cells is a lot of work and expense. And so in our ADA skid work, again, we have a lentiviral vector. We've treated now close to 40 patients with it, and 38 of them are essentially cured with it. But to get over that last hump to FDA approval might be $25 million to do that work to demonstrate you can make it consistently. And so that's the biggest bottleneck for gene therapy currently. The technology has improved, and we can do lots of things, but to get it FDA approved so you can charge for it and get reimbursed is a real challenge. So only a few have been approved. In the US, I think there's 4 stem cell-directed gene therapies that are approved for sickle cell, thalassemia, adrenoleukodystrophy, and metachromatic leukodystrophy. And there are, a couple of dozen where we have good data, but no one's been able to get it over that mountain of getting to FDA approval yet. So it's an endless source of discussion. How can we do this? So let's say it costs $25 million to do that, and you sell the drug for $2 million, which is a typical price, which is higher than it should be. So, and your costs, let's say, are $1 million, you'd have to treat, 4 or 5 years worth of patients to just recover that initial investment. So it doesn't work well in a capital model.
Right.
Investors want return on investment. And so we're looking at other models. There's something called a public benefit corporation, where you're a corporation that can make a profit, but your mission statement isn't to maximize profit, it's to make the drug available and charge enough that you can make the next batch, et cetera. And so there are possible ways to do it. I went to medical school, I didn't go to business school, so I'm not, but I'm learning phrases like fiduciary responsibility for maximizing return on investment. And so it's a real problem across the field. Jennifer Puck is here. They've made a lentiviral vector for Artemis skid, great results, kind of stuck in the same place. Really good early data, but to get from there to FDA approval is a big challenge. And so, I think it'll take all of us to put pressure on Congress or FDA to find a mechanism to allow for rare diseases. And besides, I'm talking about immune deficiencies and blood cell diseases. There's, 30 times more neurologic diseases and other things where there are other gene therapies, again, that could be brought to clinic or are in clinic already, but to get that last step to get approval so you can get reimbursed. And then actually one more step, after the FDA approves you, that doesn't mean the insurance companies are going to agree to reimburse it. So you have to then convince them. And, basically with ADA skid, the patients are getting enzyme injections that are about $300,000 a year. So we can make the argument, if you pay for this auto transplant, in a year you're going to break even and start saving money compared to if you're paying for enzyme all the way through. And in a bigger sense, if you treat the patients at a year of age with gene therapy and they're fine after that, the lifetime savings in medical costs as well as their well-being is worth it, but you have to convince insurance companies. And countries with single payer, I think it's a little more obvious. They can recognize, okay, we're going to put it out this year, but we'll save it over the next decade. In the U.S., with people switching insurance companies, et cetera, it's a little more diffuse to recognize the benefits. So there are, you know, we always just thought about the science, but in fact, it's not that simple.
Yeah, sounds like layers of complexity.
Yeah.
Yeah, thank you.
I think that's it. We're out of time for questions. Thank you so much for your valuable questions, and thank you, Dr. Cohn.